Hello, Assalamu alaikum everyone. This is now the 10th lecture on the topic of thanatology. We are discussing hypostasis, and in this lecture we'll discuss the internal findings in the hypostasis, then the progression and how much time it takes to fix on, then the medico-legal significance or medico-legal importance of the hypostasis, and what are the conditions which simulate, that is the differential diagnosis which simulates with the hypostasis, that is the congestion, bruise, and cyanosis. So we will discuss these changes in relation with the hypostasis. Thank you. Take care. Hello, everyone. Now I'm starting with the 10th lecture of the thanatology. We are discussing Bismillahirrahmanirrahim. We are discussing the hypostasis, and in this 10th lecture, we will discuss the internal autopsy findings in hypostasis, that the development of or the appearance of hypostasis internally on the organs, and the factors which will be affecting in the development of hypostasis or post-mortem lividity. Then we'll discuss the time of appearance and the fixation and the medico-legal importance of hypostasis. Then the differential diagnosis of lividity, that means the changes or the conditions which simulate hypostasis, that is cyanosis, congestion, and bruise. These are the conditions which simulate hypostasis, so they should be differentiated at the autopsy. Then we'll discuss the graphic representation or graph appearance of the hypostasis, not bruise. This is mistakenly written. Graphic representation of hypostasis. So regarding the internal appearance of the hypostasis, the distribution of hypostasis in the viscera is also dependent upon the position of the body, that in which the body is lying internally on the viscera and the organs, the hypostasis will develop on the dependent areas of the viscera. When the person is in supine position, the hypostasis will be seen in the posterior portion of the cerebrum, posterior portion of the cerebellum, dorsal portions of the lungs, posterior wall of the stomach, dorsal portion of the liver, kidneys, and lowermost coils of the intestine, which are dependent. So it is important to differentiate lividity from other congestive conditions or congestive changes which appear because of some pathology before death. So we should distinct them from lividity. As in case of lungs, this hypostasis should not be mistaken for inflammatory congestion of pneumonia, because the congestion of pneumonia will be developing on all the surfaces of the lung, not only limited to the dependent area or dependent part of the lungs. So the hypostasis, similarly in stomach, it should not be taken as engorgement due to some irritant poison, because in that case also the congestion or irritation will be on all the surfaces, not limited to the dependent region or dependent area of the stomach in which the hypostasis develops. So if there is some confusion, histology can decide this issue, because in hypostasis, the blood will be intravascular. it will not be outside the vessel, and there will be no inflammatory cells. Like in inflammatory conditions or infections, there will be cells of inflammation or infection in the histology section. So this is the lung showing the hypostasis. You can see the dorsal surface, which is showing the hypostasis. Then what are various factors which affect postmortem lividity? The postmortem lividity is affected by, as it is a postmortem phenomena, and it is due to reduced hemoglobin. There is fluidity of the blood initially, and because of the effect of the gravity, it will flow down to the dependent region. Then the color of the blood will be normal, and as in normal cases of deaths. Similarly, the color of skin will be normal, and ante-mortem state of body and mode of that should be differentiated from the post-mortem lividity. The position of the body and the pressure effects on the specific areas because of tight clothing or the some protruding object, that should be also kept in mind. In carbon monoxide poisoning, it is bright cherry red color because carbon monoxide has many hundred times affinity than oxygen, and it it doesn't allow the oxygen to take part in the metabolism, and blood remains cherry red color. Then in acute cyanide poisoning, it is bright pink color, which later fades away. And this is due to interference with the cytochrome oxidase enzyme, which is needed for the cellular metabolism. In potassium chlorate poisoning, the color of the staining is chocolate brown color due to formation of methemoglobin. In death due to cold, as in extreme hypothermia, optimum temperature for metabolism is required, but in lower temperature, the metabolism will not be going on and oxygen will not be utilized, and the blood will remain brighter. So bright pink color is due to hypothermia. Then in hemorrhagic or anemias, there is faint traces of postmortem staining. As due to hemorrhage, the blood has been lost and very little amount of blood is available for development of postmortem staining. So faint marks of hypostasis will be seen in conditions of hemorrhage. And in asphyxia, it is dark purple color because of deoxygenated blood. Then the time of postmortem lividity and its fixation. In about 20 to 30 minutes after that, it appears as small mottled patches. In next 2 to 3 hours, they coalesce together and become broader patches. And this is completed in 6 to 8 hours. In 6 to 8 hours, it is completely developed. And in about 8 to 12 hours, it is fixed. Fixed means now it will not change its position. It will remain organized or developed in that areas. It will not now shift. If the body is shifted in this position, it will not reappear in the new dependent areas. But if it is position of the body is changed before the staining has been fixed, it can or it may reappear on the next dependent areas. So the test of fixation is that you just press the areas with the thumb of your hand like this. You press the area, and if it bleaches, that means postmortem staining is not fixed. And if it doesn't bleach, that means the postmortem staining has fixed. This is a graphical representation of lividity. You can see it starts a few times after death, and in about 8 hours it is fully developed. Then it stays for next 24 hours after development till the putrefaction sets in, then it will disappear in next few hours. Now, regarding the medico-legal importance, it is a reliable sign of death, and it tells about the position of the body at the time of death. The initial position and the later position, whether it has been changed or not changed, it tells us about this. Then the color suggests about the cause of death, because the color of the hypostasis is the color of the blood. Then it helps us in determining time since death. In addition to other signs of death, the hypostasis can also be considered as important sign of death, and lividity has not much role in determining time since death, but still we can have a rough idea about the time since death, because as we know, it takes 20 to 30 minutes to start in. It appears initially in small bladder, small patches. Then in next 2 to 3 hours, the small patches coalesce together and they become broader patches. And in six to 8 hours, it is fully developed. So we can see in which stage it is of development, and we can estimate the time since death. Then there are certain conditions which simulate differential diagnosis of lividity and other conditions like BRUISE, congestion, cyanosis. We will discuss these. So in carbon monoxide poisoning, it is bright cherry red when the carboxyhemoglobin concentration is over 30%. In opium poisoning, it is black. In extreme cold, it is reddish pink. Potassium cyanide, it is pinkish. And this is the postmortem staining of carbon monoxide. This is in opium poisoning. Now, the medico-legal importance regarding the position of the body at the time of death. It shows the position in which the body remained after death, so we can have an idea about the manner of death, like hanging, drowning, and other conditions. When the position of the body is changed before the pattern of lividity is established, fresh areas of lividity will develop in the new dependent areas, but faint patches of lividity will persist in the previous dependent sites. So when the position of the body is changed after distribution pattern of the blood in the dependent vessels have been fixed, the pattern of lividity cannot be altered. This is from Shapiro. And fixation of lividity is due to stagnation of blood in the distended toneless blood vessels, and it is only a relative term. When a body is turned or its position is changed, same of it will have to fade out. Well-developed hypostasis, however, fades slowly. But if it has been organized or fixed, it will not change its position. So therefore, if a body is found in a certain posture with the postmortem lividity distributed over a region of body which is not the most dependent part of that posture, then it can be assumed that the body has been moved or its position has been changed after death, after fixation of the lividity. Now, regarding the differential diagnosis of postmortem lividity, that is the conditions which simulate lividity. They are cyanosis, BRUISE, and congestion. These three conditions simulate postmortem lividity. Lividity and cyanosis. We'll discuss the contrasting features in both. This is cyanosis. This is tips of the fingers and nails are cyanosed. So lividity and cyanosis. The postmortem lividity is a postmortem phenomena, whereas cyanosis is an antemortem phenomena. The lividity is on dependent parts, whereas the cyanosis develops on terminal or the tips. Then appearance, it is normal in all types of death in postmortem lividity, whereas cyanosis is abnormal pathological finding. The confirmation, the visible as postmortem phenomena, whereas the cyanosis is antemortem phenomena, and other evidences can be correlated with this finding. Then the change of position, if the postmortem staining has not been fixed, it will shift, whereas cyanosis position will not be changed. Then there is history of disease in the cyanosis, whereas there is no relation of any disease in postmortem lividity. Then the confirmation that it is a postmortem phenomena and cyanosis is an antemortem phenomena. Now lividity and the bruise. The lividity, as it is a postmortem phenomena, it is on the dependent regions of the body, and it is due to filling of the toneless blood vessels. Whereas in bruise, it is subepidermal due to rupture of the vessels because of trauma, and the blood has been extravasated out of the vessels. Whereas postmortem lividity, the blood remains within the vessels. It does not come out of the vessels. Then the cuticle in postmortem lividity is uninjured, whereas in bruise, it may be injured. Then regarding the location, the postmortem lividity develops on the dependent parts of the body in which it is lying, whereas the bruise can be on any site of injury at which the weapon has been inflicted. Now the appearance, it as the postmortem lividity is the pooling of blood because of gravity, there is no sign of inflammation or swelling, whereas the bruise, there will be swelling due to accumulation of the blood and the edema fluid. The edges in postmortem lividity, they are clearly defined, but bruise, they merge with the surrounding areas of the tissues. Then the color is uniform in postmortem lividity, whereas bruise, the color is variegated because the hemoglobin has been subjected to disintegration, and in various stages of healing will be showing different colors of the bruise. And in cut section, when we incise the postmortem lividity area, the blood flows from the vessels and it can be easily washed, whereas when you incise the bruised area, blood has been extravasated out of the vessel, it has been organized into the tissues, and it cannot be washed. If you try to wash it, it will not wash away. Then the effects of pressure, the postmortem lividity, it will not develop on the areas which are under pressure because of tight clothing or because of some protruding object pressing the body. But bruise will be appear under the pressure areas. Then the blood elements, if you do the microscopy, the blood elements seen in the blood vessels with no evidence of inflammation in the postmortem lividity, whereas the blood elements will be seen outside the blood vessel with the evidence of inflammation in case of bruise. This is the cut section. There is no vital reaction. This is the postmortem lividity, and the blood can easily be washed. So these differences will be marked before the putrefaction starts. But when the putrefaction sets in, then it will not be differentiated. An important thing is that the putrefaction starts relatively earlier in places of bruises. So this is the lividity on the dependent regions. And this is bruises. You can see even on the front of the body and side of the body. This is bruise on the back of the hand. And this is showing a variegated colors. The hemoglobin undergoing disintegration and various colors of healing will be seen in case of bruise. This is also showing the variegated colors. Now, lividity and congestion. The lividity is dependent or distributed on the dependent parts, where the congestion involves the whole organs. Then appearance is normal in case of postmortem lividity. It appears in all types of death, but it is pathological change in case of congestion because of abnormality. Then the mucous membrane in postmortem lividity will be dull and lustrous, whereas in case of congestion, it will be normal. Regarding the exudates, no inflammatory exudates will be in postmortem lividity, whereas in congestion, the inflammatory exudates will be seen. The hollow viscera, the postmortem staining will be developed on the dependent regions, and when the organ is stretched, they will be altered, that because of the stretch the blood will flow, but the congestion it will be uniform distribution, and because of stretch it will not be uh, alter its state or alter position. So that's all about this lecture, and the summary of this lecture is that we have understood what are the internal appearance of various organs in hypostasis, and what are various factors which can affect the postmortem lividity, the time of its appearance and fixation, and medico-legal importance of hypostasis. And what are the conditions which simulate hypostasis, that are the differential diagnosis of hypostasis. That is cyanosis, congestion, and bruise. We have learned about how they are distinct from the lividity. Then this is again a typic mistake. We have seen the graphic representation of lividity, not bruise. This is mistake, typing mistake. Thank you very much. That's all about the 10th lecture. Take care. Allah Hafiz.